Hello everyone. My topic is mechanism of insect hormone action. So first let's have a quick introduction. In insects, hormones play crucial roles in various physiological processes. Juvenile hormone regulate metamorphosis, molting and reproductive development, while ecdysteroids control molting and metamorphosis. Additionally, neuropeptides influence behaviors and physiological functions. So here we could see different insect hormones. Juvenile hormone is there, ecdysteroids and neuropeptides, insulin-like peptides, allotostatins, then coraxonin, prothoracicotropic hormone, diuretic hormones, iron transport peptides. So, hormone action in insects refers to the physiological and developmental effects exerted by specific chemical messengers known as hormones on various target tissues and cells within the insect's body. Same is the case with humans. These hormones play a crucial role in regulating essential processes such as growth, development, metamorphosis, reproduction, and behavior. The action of hormones involves a series of steps, including synthesis and release of hormones from specialized glands, transport through the circulatory system, hemolymph through hemolymph, then recognition by target cells through specific receptors, and subsequent signaling cascades within the cells leading to specific physiological responses. Next, mechanism of hormone action. Hormone action involves a precise sequence of events. First, hormones are synthesized by specialized glands or tissues. They travel through the bloodstream to target cells where they bind to specific receptors. This binding activates intracellular signaling pathways, leading to a cellular response, such as changes in gene expression or enzyme activity. Then, different steps of this process: synthesis and release. Hormones are produced by endocrine glands or specific tissues in response to physiological signals or external stimuli. That is the first step. Then transport will be taking place. Hormones travel through the bloodstream or other circulatory systems to reach target tissues or cells. Then recognition. Target cells possesses specific receptors that recognize and bind to the hormone molecules. This interaction is highly selective, like a lock and key model. Then signal transduction will be taking place. Hormone binding activates the receptor, initiating a cascade of intracellular events. This often involves second messengers or alterations in the activity of cellular proteins. Then the first step in mechanism of hormone action in insects is the synthesis of hormones. Specialized glands or tissues within the insect's body produce these hormones in response to various signals, including developmental cues or environmental factors. For instance, in insects, juvenile hormones are synthesized and released by the corpora allata gland, and ecdysteroids like ecdysone are produced by the prothoracic glands. The synthesis initiates hormonal cascade that regulates crucial physiological process in insects. So, juvenile hormone is synthesized and released by the corpora allata gland, and ecdysteroids like ecdysone are produced by the prothoracic glands. After synthesis within specialized glands, insect hormones are released into the hemolymph, the insect circulatory system. This release is prompted by developmental cues or environmental signals. Traveling through the bloodstream, hormones reach target cells where specific receptors await in the cell surface. The binding of hormones to these receptors initiate a cascade of intracellular events, including signal transduction pathways. Then signal transduction cascade, the binding of the hormone to its receptor, sets off a series of events within the cell. This often involves the activation of second messengers such as cyclic AMP and calcium ions, and the modulation of protein kinase activity. These intracellular signaling pathways transmit the hormonal signal to the cell's interior. Then gene expression regulation will be taking place. The activated signaling pathways can influence gene expression. This means that specific genes within the cell may be turned on or off, leading to changes in the synthesis of proteins and other molecules. Then cellular changes will be happening. The altered gene expression and protein synthesis result in specific cellular responses. This could include changes in cell structure, metabolism, or the activation of particular enzymes. So this could include changes in cell structure, metabolism, or the activation of particular enzyme. Physiological effects will be taking place. The cumulative effect of the cellular responses manifests at the physiological level. For example, in insects, the actions of hormones may regulate molting, metamorphosis, reproductive process, and behaviors. These physiological effects will be happening. Then feedback mechanisms are there. Throughout this process of enzyme action, feedback mechanisms operate to maintain balance. Excessive cellular response may trigger feedback loops that regulate the hormones production or signal transduction pathways. Feedback regulation, the cellular responses triggered by hormones often feed back to the glands responsible for hormone synthesis. This feedback can be negative where the cellular response inhibits further hormone production. There will be a stop switch kind of mechanism where the cellular response inhibits further hormone production, or positive where it stimulates hormone release. Negative feedback is particularly crucial in maintaining homeostasis and preventing excessive hormonal stimulation. So, in summary, the cellular response phase represents the culmination of the hormone action mechanism, translating molecular signals into tangible changes at the cellular and physiological levels. And on of such mechanism or the feedback loops will be there. This orchestration ensures that hormone regulation is finely tuned to meet the organism's needs and maintain internal balance. Adjustment to hormone levels. Based on the feedback received from the cellular responses we mentioned, the glands adjust the production and release of hormones. If the cellular response indicates that the physiological needs have been met, hormone synthesis may be reduced. Conversely, if the response suggests a deficiency, hormone production may increase. And homeostasis maintenance, the intricate interplay of feedback mechanisms ensures that the level of hormones in the circulatory system are maintained within a specific range. This balance is essential for the proper functioning of physiological processes and preventing overstimulation or understimulation of target cells. So, feedback mechanisms are very important, and this specific range of hormones are present that will help in the prevention of overstimulation or understimulation of target cells. Adaptation to changing conditions. The feedback mechanisms also enable the insect to adapt to changing environmental conditions. For example, in response to seasonal changes or stressors, the regulation of hormone levels allows the insect to adjust its physiology and behavior accordingly. So, this environmental regulation is also according to environmental conditions, regulation is happening inside this insect's body. That thing is also happening. Now let's learn factors affecting hormone action. Hormone concentrations. The concentration of hormones in the circulatory system influences the strength and duration of the hormonal signal. Fluctuations in hormone levels can result from factors like developmental stages, environmental cues, or stress. Hormone concentrations are very important. And feedback mechanisms we mentioned. Negative feedback loops play a vital role in maintaining hormone homeostasis. These mechanisms regulate hormone production based on the cellular responses, preventing excessive stimulation or inhibition. Then environmental factors, external conditions such as temperature, photoperiod, and food availability can influence hormone levels and action. Insects often use hormonal pathways to adapt their physiology and behavior to changing environmental conditions. Then stress and environmental challenges. Environmental stressors in case of insects such as predation, temperature extremes, or exposure to toxins can trigger hormonal responses. These responses help insects cope with and adapt to challenging conditions. And this have a considerable amount of impact on hormone action. And genetic factors, variations in the genetic makeup of individuals can influence hormone receptor expression and function, leading to variability in the responsiveness of hormones. And hormone initiate intracellular changes in target cells with appropriate receptors. So receptor proteins in target organ determine the specificity of the physiological response. Now we are considering some general important points related to this topic. So factors affecting hormone action we mentioned. Now some general points. Receptor proteins in target organ determine the specificity of the physiological response. Not all cells respond uniformly to hormones. So receptor proteins presence and variation contribute to differential cell responses. So these are some basic aspects related to hormone action. Only cells with suitable receptors are affected by a specific hormone. There is some sort of specificity between these cells and their suitable receptors for a particular hormone. So receptors may exist in different forms and are expressed variably. And depending on the hormone type, receptors may be in the cell membrane or within the cell. So first we are going to elaborately learn about ecdysteroids as an example. So ecdysteroids are of lipophilic nature. Ecdysteroids are assumed to be lipophilic, facilitating easy passage through cell membranes. Then cellular uptake, limited understanding of the process despite attempts to characterize the cellular uptake of ecdysteroids. Intracellular binding. Once inside the cell, ecdysteroids bind to specific receptor proteins located in the nucleus. Then lipophilic nature is the nature of the ecdysteroid. Then cellular uptake, limited understanding of the process despite attempt to characterize the cellular uptake of ecdysteroids. Cellular uptake, limited understanding of the cellular uptake of ecdysteroids. But intracellular binding will be taking place once inside the cell. Ecdysteroids bind to specific receptor proteins located in the nucleus. Gene activation will be happening. Ecdysteroids activate a small number of genes directly, influencing the synthesis of molting related proteins. Then receptor structure is very important. DNA binding region. Both subunits of the receptor contain a DNA binding region with zinc fingers, typical of nuclear receptor DNA binding transcription factors. Here we mentioned different types of insect hormones in the first stage. Now we are selecting different hormones, insect hormones individually, and analyzing their mechanism of hormone action. So in case of this hormone, DNA binding region is there. Both subunits of the receptor contain a DNA binding region with zinc fingers, typical of nuclear receptor DNA binding transcription factors. Then recognition of ecdysteroids. The E region of ECR recognizes the presence of ecdysteroids, but binding requires the presence of USP. Then ecdysone response elements, ERE region. The heterodimeric ecdysteroid receptor binds to specific regulatory DNA sequences or ERE near molting related genes. Then transcriptional regulation will be taking place. In the absence of molting hormone, ECR associated with proteins like SMRTER, SMRTER, represent target gene expression. Don't forget the spelling of this protein. Then that protein is associated with the molting hormone. In the absence of molting hormone, this ECR is associated with proteins like SMRTER, SMRTER. Yeah, this SMRTER protein is present when molting hormone is absent. So RER is associated with SMRTER when molting hormone is absent. That will help in the repressing activity of target gene expression. Then presence of ecdysteroid causes a change in the receptor's molecular shape, leading to transcriptional stimulation. Then transcription activation function, AF. ECR has two AF regions, AF1 and AF2 regions are there, contributing to transcriptional activation. Then AF1 in the A by B domain and AF2 in the E domain play roles in promoting transcription. This is some technical aspects or some important points related to this. So deletion of AF2 results in a dominant negative receptor. I don't think when studying for exam this much information is not needed, but I want to just convey the message with you. So deletion of AF2 results in a dominant negative receptor. Then ecdysteroid regulation of gene transcription. Early response genes. Ecdysteroids regulate the transcription rate of early response genes, which early response genes are shown here like E74, E75 and Broad complex. And transcription factors. These early response genes encode transcription factors that play a crucial role in the molting process. Early proteins are late genes. Early proteins derived from the early response genes regulate the expression of a larger number of late genes involved in molting events. Stimulation and expression. Expression of molting related genes is stimulated while some are repressed, with relief from repression when ecdysteroid levels decrease. Molting and metamorphosis coordination, direct and indirect gene regulation through ecdysteroids leads to a complex time and tissue specific pattern required for coordinating molting and metamorphosis. In early studies in diptera insects used the perfect pattern of polytene chromosomes to indicate transcriptional activity. Modern techniques like gene chips provide detailed qualitative and quantitative insights into transcriptional control. An mRNA regulation is also needed. Ecdysteroid surge towards molting results in the rise or fall of large number of mRNAs, indicating the broad impact of gene expression. Tissue specificity and timing. Tissue specific and timely response to ecdysteroids are influenced by other transcription factors present in cells. And ECR protein variants. The ECR protein exists in different variant isoforms A, B1, B2 in Drosophila due to varied post-transcriptional processing. And ECR mRNA splicing and isoforms. Splice variants ECR and mRNA undergo splicing, resulting in different splice variants of ECR protein. And tissue development expression. Splice variants are expressed differently in various tissues and at different stages. Isoform differences. Isoform vary in A by B region, influencing their ability to promote target gene transcription. ECRA lacks AF1 function, affecting its role in promoting transcription. And target gene expression. Different ECR variants have varying abilities to promote the expression of specific target genes. And developmental importance. The pattern of expression of ECR isoforms holds potential importance in development, although specific roles are yet to be fully understood. Alternative actions via signaling pathway. Some actions of ecdysteroids, like apoptosis of Bombyx anterior silk glands, involve a different and faster acting signaling pathway. This pathway utilizes cell surface G protein coupled receptors or GPCRs. Next is juvenile hormone action and receptor. Morphostatic action in immature insects. JH morphostatic action in immature insects involves modifying transcriptional response to ecdysteroid molting hormone, maintaining the status quo. Requirement for cellular entry. Despite the common belief that JH has no effect by itself, it must enter the cell to modify transcriptional response similar to ecdysteroids. Then hydrophobic nature and cellular entry. JHs, being highly hydrophobic, are presumed to enter target cells by diffusing through the plasma membrane. Unknown intracellular receptor. For many years, the identity of the intracellular receptor to which JH must bind inside the cell remained unknown. Juvenile hormone action and receptor. Here we can see ecdysteroid receptor proteins are shown. Here are ECR1B1, ECRB2, ECRA, USP and USP regions. Ecdysteroid, GPCR, non-genomic effects. Then it activates co-repressor protein, early gene, transcription factor, molting, late gene. Genetic approach for receptor identification. Genetic techniques proved more successful in identifying the intracellular receptor. The Drosophila melanogaster gene, methoprene tolerant MET, was identified through mutations conferring resistance to the JH mimic insecticide methoprene. MET as a transcriptional regulator. MET include a transcriptional regulator belonging to the BHLH pass family, acting as a functional JH receptor. It binds radio-labeled JH3 with nanomolar affinity and regulates transcription of non-target genes in the JH-dependent manner. Attributes of functional JH receptor. A functional JH receptor like MET should exhibit binding of affinity for JH and regulate transcription in response to JH. Methoprene resistance and malformations. Mutations in MET confer resistance to the JH mimic insecticide methoprene, which includes lethal malformations by disrupting metamorphosis. Significance of BHLH pass family. METs belong to the BHLH pass family highlights the importance of this family of transcriptional regulators in JH action. Nanomolar affinity 4. JH3. MET's ability to bind JH3 with nanomolar affinity emphasizes its role as a specific and sensitive JH receptor. MET loss of function and fertility. Loss of function mutations in the MET gene reduces adult female fertility but are not lethal in Drosophila, suggests that MET mediates specific actions of GH or shares its functions with at least one other receptor protein. Germ cell expressed as another JH receptor. The GCE gene encoding a PHLH PAS protein closely related to MET is identified as another probable JH receptor in Drosophila. Conservation across insects. Many insects have only one gene of this type closer to GCE than to MET. Implies that a GCE-like protein may mediate morphostatic JH actions in insects beyond Drosophila. Then heterodimer formation, MET, GCE and FISC. Initial suggestions of MET and GCE forming a heterodimer as a JH receptor. Currently, fish, a third DHLP PAS protein, seems a more likely candidate as the heterodimeric protein of MET. Then JH influence of the JH influence of gene expression. Once JH binds to its receptor, it influences the expression of genes affecting development. Interplay with ecdysteroids. For morphostatic actions of JH, responsive genes are also influenced by ecdysteroids. Uncertain if JH and ecdysteroids act on the same early genes or if their effects converge downstream in both regulatory pathways. JH response element JHRE. A JH response element JHRE identified in mosquitoes is crucial for JH dependent gene expression. The MET FISC complex binds to this DNA sequence in vitro, supporting evidence of a heterodimeric nuclear receptor, likely including GCE or GCE-like protein and FISC. Involvement of ultraspiracle USP, the heterodimeric partner of ECR in the ecdysteroid receptor, binds specifically to labeled methyl farsenonate. We mentioned this USP in this figure. Yeah. MET as a transcriptional regulator. MET encodes a transcriptional regulator belonging to the BHLP PAS family, acting as a functional JH receptor. Methoprene resistance and malformations. Mutations in MET confer resistance to the JH mimic insecticide methoprene, which induces lethal malformations by disrupting metamorphosis. Significance of this family. METs belonging to the BHLP PAS family highlights the importance of this family to transcriptional regulators in JH action. Nanomolar affinity of JH3. MET's ability to bind JH3 with nanomolar affinity emphasizes its role as a specific and sensitive JH receptor. MET loss of function of fertility. Loss of function mutations in the MET gene reduce adult female fertility but are not lethal in Drosophila. Suggests that MET mediates specific actions of JH or shares its function with at least one other JH receptor protein. Germ cell expressed another JH receptor. JH response element. A JH response element identified in mosquitoes is crucial for JH dependent gene expression. Complexities in JH action and USP's role. Uncertainty exists about the physiological relevance of methyl farnesonate in Drosophila as a JH-like product. Challenges reconciled findings on USP, MET, GCP, suggesting that both schemes may be too simplistic. Potential interaction of JH receptor and USP. Possibility that the JH receptor and USP may cooperate in some JH actions. Independent JH actions in adult insects. In adult insects, JH can be independent of ecdysteroids, influencing sexual maturation and behavior. JH stimulates a protein kinase activating an ATPase in the cell membrane, resulting in osmotic challenges, cell shrinkage, and the appearance of spaces between cells. Juvenile hormone, JH responsive genes, transcription factor, then morphostatic effects are there, ecdysteroid responsive gene, GPCR kind of reaction, and ATPase. Next is peptide hormones and biogenic amines in cellular signaling. Lipophobic nature and cell membrane interaction. Peptides and amines are primarily lipophobic and do not pass through cell membrane. Specific receptor proteins in cell membranes interact with these hormones to initiate signaling cascades. Activation of second messenger synthesis. Peptides and amines activate cascades of second messenger synthesis and of protein phosphorylation via their specific membrane receptors. Intracellular enzymes usually present but inactive are activated through these cascades. Receptor proteins and gene protein coupled receptors. Many membrane receptors belonging to the GPCR superfamily with 7 membrane spanning domains. Then GPCRs associated with heterotrimeric GTP binding proteins. Upon hormone binding, a conformational change releases GDP from the G protein replaced by GTP. The common GPCR mechanism. G protein activation and intracellular affects the G protein's alpha subunit along with bound GTP, dissociates and interacts with target protein in the cytoplasm. Effects depend on the type of alpha subunit affecting rate limiting components controlling specific intracellular second messengers. Examples of GPCR signaling pathways like GS activates adenylyl cyclase producing CAMP from ATP, leading to immediate cellular response via protein kinase A. CAMP also initiates long-term responses by activating the CAMP-dependent transcription factor CREB. Alternative GPCR-linked signaling pathway GQ activates phospholipase C, cleaving membrane-associated phospholipids. This produces second messengers inositol triphosphate IP3 and diacylglycerol DAG. IP3 promotes the release of Ca2 plus ions into the cytoplasm, leading to cellular responses via calmodulin and protein kinase C. Intracellular mechanisms of insect peptide hormones. Knowledge of intracellular mechanisms for insect peptide hormones lags behind structural and receptor identification. Experimental caution is necessary due to varying hormone effects in different insects, stages of development, and potential simultaneous multiple cellular actions. This is a picture. Cellular signaling of adipokinetic hormone and other insect hormones. Adipokinetic hormones or AKH action are mediated by a single type of GPCR related to the mammalian gonadotropin releasing hormone receptor. The receptor can be coupled to multiple cellular signaling pathways influencing various responses. Then diverse responses to AKH, mobilization of lipid or proline in different insects results from AKT acting on fat body cells via GS, causing elevation to CAMP levels and increased intracellular Ca2 plus levels. Mobilization of stored carbohydrates in some insects involves DAG and IP3, prompting Ca2 plus release from intracellular stores. Downstream targets and uncertainties. The downstream targets for AKH may vary in fat body cells of different insects. Uncertainty whether different responses are due to the presence of only GS or GQ in fat body cells, or if both signaling cascades are present with certain downstream targets absent. Then pheromone biosynthesis activating neuropeptide. PBAN's actions like AKH are mediated by a single GPCR type, but different insects utilize distinct cellular signaling pathways. Fluid transport regulation in Malpighian tubules. In various insects, fluid transport in Malpighian tubules is regulated by peptide hormones from different classes. These hormones operate through different GPCR types and GQIP3, DAG, and GSCAMP pathways influencing ion and water movements. Peptide hormones with non-GPCR receptors. Some peptide hormones such as PTTH and insulin-like peptides associate with non-GPCR receptors, activating distinct intracellular pathways like MAP kinase and M13 kinase. Complex interactions in cellular signaling. Cellular signaling pathways are rarely independent, making it challenging to identify a primary route. Crosstalk between pathways is common. For example, CAMP and CGMP signaling influence intracellular calcium levels, impacting IP3 levels and creating feedback loops. Interactions may be time dependent, resulting in oscillatory changes in cell states in response to a single hormone or multiple hormones. Now let's have a quick summary. Numerous hormone substances control the physiological, developmental, and behavioral activities of insects. These belong to three major physical classes like steroids, molting hormones, sequiterpenes, juvenile hormones, and polypeptides. Hormones are secreted by specialized glandular tissues in various body regions and by modified neurons, neurosecretory cells. Regulations of hormone titers involves changes in synthesis, release, activation, degradation, and excretion. Hormones act upon target tissues via cell membrane and nuclear receptors, eliciting cellular responses and changes in gene expression patterns. Thank you for listening.